thank you for the introduction. I brought my own introduction slides. So my name is Franziska. I'm the first employee at Cradle, and we are a company who try to help other companies make better proteins using machine learning. I'm going to talk to you today about a system walkthrough of what we do, so like the whole front-to-end setup of our generative AI setup. A brief note on the team, so why I actually joined Cradle is because it's an amazing team of lead experts in biology, machine learning, and design. Especially the designers like to show their skills by making really awesome slide decks. Okay, so let's get into what we do. At Cradle, we make variants of proteins. This means our users tend to have a protein that they work with, and we apply small mutations to increase a target property. For instance, thermostability, if you want to increase the temperature in your organism or get more product, if you have an enzyme use case. How these protein projects usually look like is you run over time, so you design a set of variants, you go test them in the lab, you get results, and you build another round on the best candidate from your last round. You do this iteratively, ideally until you reach your target. Now, this process is very time-intensive and expensive. A round usually runs in weeks or months. I think the longest we've seen is 6 months. And this costs about something between 10 and hundreds per data point to evaluate a label. We try to make this process even faster so that our customers reach this goal faster. And we also try to build a generalizable platform that is able to deal with any protein and a variety of properties to optimize for. Now let's get into this. We build a generative AI system for proteins, and we'll start kind of easy. This is a slide you may have seen today already. So you have a protein sequence, which is just a string. You mask out some positions. In that case, you can just use a mask language model, ask it to fill in amino acids. You do that a bunch of times, which gives you a set of variants. That's a very easy generative AI system. But we actually want sensible protein or suggestions for mutations in that protein. So we apply a technique which has also been mentioned, which is called Evo tuning. You work with an unlabeled data set of sequences, which is called the MSA, and we actually build an MSA around our target protein to further fine-tune the big model in that case. So the big language model is being further trained to the local landscape of this problem. What I forgot to mention is, I guess, like the models, public common architectures we use are, for instance, ESM2. You can get them online, and we further fine-tune this to get our EvoTune model. Now we have a great unsupervised trained model, and if we put in more data, we can make it even better. Not sure. We actually saw that unsupervised training at some point kind of converges in its performance. So what you can see here is 3 examples of actual proteins where we tried different subsets of unsupervised training sets. The dark blue one being a combination of the other 2, and it's not a whole lot better in performance. So this kind of peaks out at some point. What you also can see is that Evo tuning in itself works pretty great. So without any label, you can reach Spearman correlations of up to 0.4, 0.5, which is quite neat, actually. So now we're at the point where we actually need to bring in the data from the lab. So a lab, they have historic data or just last round data that we use to train a simple regression model. It's called a fitness predictor, so it takes in a sequence and it predicts a performance score. With these 2 models, we put them together, we have variants, we rank them, we pick the top ones. Great, right? Actually not yet, because if you then go and look into lab data, you will notice that things are not quite as beautiful as you want them to be. So here I have a plot of actual data from the lab. The colors are different rounds. On the x-axis, you see different variants. So on the y-axis is the assay score from the lab. And you can first see that we have a lot of replicas. Second, we can see that rounds do not align with each other. And third, that it is kind of noisy process. So we decided to only require sequence sets to be consistent within a round. So one round needs to be consistent, at least in its ranking. And we call this lab round constraint training. It just means that when you train a model, you make sure that whenever you sample a batch for training, it will be part of one round where the consistency constraint is kind of given. So the model will never see sequences together from different rounds that may not make sense in that context. Cool, so now the model is also even noise agnostic. That's it, right? You build a robust system that can build better proteins. Also not there, so the last thing we add on is actually targeted towards generalization, because we run models over multiple rounds. That means a model is always working kind of the edge of its learn capabilities, because you're always taking the best sequence, you're making mutations to it again, and you're asking the model to only predict in that very local landscape of your best sequence. And this deteriorates over time, especially if you get further away from what the model has learned. You can see this here. The performance on the y-axis is decreasing over lab rounds because we're getting further away from our starting sequence. What you can also see here is a site as we do use ensembling, but it's just another nit here. How do we account for this? We actually introduced the concept of confidence. So in addition to a predicted performance, you will also get a confidence score that basically tells you how noisy your measurement is going to, or is, from the model. We use a technique called Monte Carlo dropout. So models are trained with dropout. And if you apply that dropout again at inference, it's basically noising the prediction. You can build a distribution over the predicted score, and the variance of the score basically tells you how inconfident your model is about its prediction. There's also in the plot, you can see that we again have single sequences on the x-axis, and the blue ones are the ones that we would filter out due to low confidence in prediction and not suggest as a variant for the lab. To summarize, we have looked at 3 parts. We have an evo-tuned model, which is the generation model that actually builds a new sequence. We have the fitness predictor, which then scores these sequences to give us a ranking. And then we have a confidence value to disqualify some sequences, or like some generated sequences, because we cannot rely on the predicted performance. These are some core parts of our system. We're constantly trying to improve them. Of course, this is not the whole story, but I would have needed more time for that. So as a last part, let's quickly look at if this actually works. And this is where it gets really cool, because we are working with real customers, we have real data, and we have seen cases where this works. So let's look at P450. There's also a case study online on this, where we ran a project with a customer. They have been running this project without us for 10 rounds, and you can see with mediocre success. So they did clearly not hit the target they were trying to achieve, and then they brought us in and ran with us over 3 rounds to get a lot better, and we did help them improve their performance by 16 times. With that, we're still evaluating the technique with other customers, and we of course also have an in-house lab to make sure it works right. That's it. Thank you for your attention.